Hello, everyone. Welcome back. Now we come to the report back session for the breakout sessions. So this morning, we had 4 breakout sessions on these topics. So what we will do in the next half hour is we will hear the report back from each session. So I'm Xuegong Zhang of Tsinghua University in Beijing. So I will start to report on breakout session 1. So the session 1 is about Atlas integration. So we had a set theme for this session, that is Atlas integration, technology, strategy, and the role of Asia. So the session was co-chaired by me and Professor Ge Gao of Peking University, also in Beijing. And we have the 5 panelists. And now I will go quickly through what we have talked about. We have talked about 7 major topics. The first 3 are about the technical and strategic challenges and possible solutions. For the technical challenges for Atlas integration, we talked about batch correction and the possibility, the risk of overcorrection. And scalability is a huge challenge for future Atlas integration because we are looking at more and more data. And annotation is a challenging task, and evaluation of the batch correction or annotation are all challenges. For the strategic challenges, we talked about the level of Atlas. For example, whether we want level at organ level or at the whole body level, whether we build Atlas for single omics or multi-omics, and whether we integrate data at raw reads level or at the expression matrix level, topics like that. And those are questions and the possible solutions. Some of the solutions are possible solutions, and some are just the directions we thought could be a solution, could lead to solution. Like borrowing information from other omics data may be helpful for batch correction and for annotation. And for scalability, we can take divide and conquer approach to divide large scale data into smaller ones. And we think there are, when we talk about batch correction and other pre-processing, we think we should be aware of what the Atlas will be used. So we coined the term application-aware pre-processing, which should be, so we should not talk about batch correction without thinking of what the data will be used. And also we think that batch correction is important, of course. However, with the new technology coming, and it's really challenging to get perfect batch correction. So we should develop batch invariant applications that are not sensitive to batch effect. And for the Atlas, we think that we should target for multi-level Atlas rather than a general kind of a one-size-fits-all Atlas. And the Atlas should have a layered structure. Annotation, it is important that we have a kind of a standardized annotation. And for the integration of data, of course, read level integration is preferred, but it is more challenging for both the legal, ethical, and technical aspects. So those are 3 topics. And the other topics, like what should be the final HCA like? Will it be a centralized database or it's multiple databases? This is challenging. We think that probably the Human Genome Project may not be a feasible model for the Human Cell Atlas project. And there has been many efforts happening in the world for integrating single cell data, not necessarily being official human cell atlas. And a human cell atlas, we should realize that it is not simply a huge collection of many single cell studies, because many of the single cell studies are not for the purpose of atlas-like references. They were probably most of them are for disease studies and other studies. So it is challenging for bioinformatics, maybe AI technology, to make good use of those data for building atlases. And for all those efforts, defining the minimal metadata for the data to be integrated is important. And we should keep in mind the application of how could the integrated atlas be used in the future for medicine or for the industry. We didn't have much time to go deep into this topic, but we have seen examples of applications already in cancer studies, in COVID-19 studies, and the applications include discovery of new or rare cell types and refining existing definitions of cell types. And we thought that a reference should be of general purpose rather than for some specific application. So as an atlas, it should be ready for a wide range of applications. And then standardized annotation is becoming more important to ensure the future applications. And we also talked about what role can Asian and Asia Pacific community play in the integration. We all realize that there is a large space for Asian Pacific community to enhance our role in HCA. And but there are practical issues, like for most of the Asian Pacific countries, most of the funding supports only disease or translational studies rather than reference building efforts. And however, most of the countries in this area put emphasis on bioinformatics and AI approaches for single cell biology, and this can be the future solution for integration of non-atlas oriented data. And we have some special disease spectrum and population structure. We should take use of that, and we think it's a good model to form some international consortium or the regional consortium for possible international grants. And we briefly mentioned about legal and ethical issues, but we realized that it turned out that there seems to be more legal restrictions nowadays on sharing data, genomic data in several nations. And a possible way to solve this problem is trying to define, or probably by our community, a clearer red line for which type or what type of data are shareable according to the legal regulations. And we end up, we came up with some conclusion that is, at the current stage, there are more questions than answers actually for Atlas integration. But clarifying and defining those challenges are equally important as solving them, if not more important. And we agree that probably we will try to summarize our discussions to form some kind of opinion or position on Atlas integration from this Asian Pacific community, and to get our voice heard. So that's the report from the breakout session 1. So now we move to the report from breakout session 2. If we have any questions to discuss, I think we will put it in the next session, the plenary discussion session. So now we will have about 10 minutes, 10-second break to let Senjuti ready online.
Hello, everybody. Thank you for being here. So I am here to summarize what we learned from our breakout session on equity. So that breakout session was led by Alex Shalik of MIT and myself. Unfortunately, it's about 3:15 a.m. in Boston right now, so Alex had to leave right after, so you have only me. I do not have slides. However, I will try to summarize everything that we learned and the lessons that we are bringing back. In our session, we had an excellent set of panelists from different countries who brought together a diverse set of experience, very valuable for a breakout session on equity. Something that we started off right at the beginning with is acknowledging that when we discuss equity, specifically in Asia, we have to remember that Asia is quite different than any other region like EU or Latin America. In Asia, we lack political unity. We are far from having a unified vision, particularly for science in this region. So if we really want to make progress and we all want to come together, it needs to be driven by scientists. It's just not going to happen through governments or policymakers at this point. Going into some specifics, we talked about ideas to unify scientists in this region, as we are the ones who have to drive this forward. And there were many questions about how do we unify scientists? Do we unify scientists based on the experiences that local scientists already have, or do we let that happen organically? And should HCA have unifying concepts and try to consolidate people into those concepts or threads? I think there were different opinions, and this is something that we still have to decide, and they're not mutually exclusive. We heard a lot of stories about how things happened in Singapore and Hong Kong, and some interest, let's say in Singapore, about liver and how people focused on that. And some of the agenda were also driven by the government. It was also emphasized that because scientists will be moving this forward, different things excite or motivate people. And there are sometimes local priorities that need to be taken into consideration. And that, of course, brings up the situation of funding. If funding is going to come from local governments, priorities need to be set up according to the priorities of the government. And oftentimes, priorities of the government are disease specific. For example, in some countries, it's not only even disease specific, it has to be like, let's say, cancer. When we talk about scientists coming together in Asia, and we take all the countries into consideration, where some countries have a lot of training or capacity for human single cell analysis and others don't, of course the issue of training comes up, because if we want to have equitable access to the technology and benefits of the population from this technology, we need to think about training and knowledge building, capacity building. We all agreed in our session that creating opportunities for equitable opportunities for all participants or countries is key. Specifically creating opportunities for people to come together and network. At several points during our conversation, a huge genomics consortium was mentioned, Phantom, functional annotation of mammalian genome, which worked really well because it pulled together many people from different backgrounds, and they were working together for days and weeks, learning or doing science together. And that was brought up as a really good example. As we were talking about Phantom, we also thought, what can be done now? And of course, being in the middle of a pandemic, having this virtual meeting and going to many other virtual meetings for many other of our fields, we talked about, can we leverage the COVID or the virtual platforms that we have created for training, for, let's say, online training? And now that a lot of infrastructure has been set up already, online trainings are definitely very important. There was no disagreement on that. However, we must, even if we can set up online trainings, we must remember that student exchange programs are very, very important. And that should be a big aim of bringing equity into HCA. Motivating young people is the most sustainable way to move forward. To exchange programs, building relationships through collaborations is going to be very important. And as we build out these training programs, wherever we start, whether online or on site in the future sometime, we must remember that if we want to be inclusive, different people or different groups, different countries start at different levels. In order to acknowledge that we want equitable access, we have to take that into consideration and perhaps have different levels of trainings. And trainings cannot be meeting based where, we get together for 1 to 2 days. It must be like for longer time where people can meet and learn to analyze data or learn some techniques that they can take back or do in their lab. So, we talked about training, we talked about what it really means to give equitable access, but then the idea, and when we talk about priorities, then the question of translation came up. Translation came up again and again and again, and translation in two different ways, right? Number one, literal translation. Translation of all the work, all the educational material, or publications, very importantly, the publications of HCA. Translation of that into local languages came up. In many countries, most countries in the world, English is not the most spoken language. Perhaps it's still spoken by majority of the academics, but it's a lot more accessible. Knowledge becomes a lot more accessible when they're translated into local languages. So, when we start a program or build educational material, we must remember that, again, if we want to make things equitable, we must be thinking about language and access to the groups we will be working with. And now we can't, if we also want to raise awareness and educate, we not only have to translate educational materials or publications, it's also important to perhaps share ideas or share thoughts to blog posts, through local newspaper articles, etc. But then the question is, where do we start, right? Where do we start our translation? Do we start our translation just with the educational material, with publications, with certain publications, with certain languages? So of course we didn't reach the answer. We just came up with a lot more questions, as we just heard also, we came up with more questions and answers like every other meeting. But we do feel that broader conversations about what we do, what we have, and how do we collate all of that for access from multiple groups is very important questions to be answered. Basically, how do we create megaphones so that we know, so that we can reach those who we want to reach. The second translation is scientific translation of our work. Oftentimes, our funds will depend, if you apply for a grant, it will depend on what is the translation of your work in 2 to 3 years. Governments will only give funds if we can show some kind of tangible translation within 2 to 3 years. Where does that leave our idea of studying healthy cells? Oftentimes, we have to collaborate with clinics to get samples, and clinicians or physicians or collaborators will have some kind of desire to know what the translation will be, how it will benefit the patient health or public health. Basic science is definitely important. We all acknowledge that in this meeting, but it's harder. It's getting even harder to get money for that. And then we kind of came back to this idea of all of these things that we discussed. How do we get everybody on board, or how do we make these conversations broader? If we talk about translation, whether we talk about training programs, whether we talk about collaboration, where do we start? We wanted to discuss a little bit about what are the systems in place already that have achieved what we are trying to achieve. We don't have to reinvent the wheel per se. There are many historical good examples, successful examples of how technology and knowledge has been spread to the broader community. We talked about WHO, we talked about CDC. WHO runs over 50 labs in about 150 countries. They have actually built this network. They have built this network to bring many labs together. They have ways of getting reagents to labs, and they have also ways of training and making sure quality is assured. No equity talk can end without, or is complete without, talking about data sharing. So we talked about equity of data sharing. What can we share? What can we not share? In the past, there have been barriers, country specific barrier. And while we are talking about all of this, funding obviously kept on coming up. Where do we get the pilot money from? What is the incentive structure? Some researchers are not connected to clinics, so they have to go to a clinic to collect samples. Where does the funding for that come from? So there were a lot of discussions, and again, no answers, but at least we realized that there is more to be discussed. To conclude, we all acknowledged that collaboration at the beginning is very important. Equitable collaboration that has bilateral benefits, building trust, building responsibility is important. And at the end, we acknowledged that, you know, we all bring our biases over here. Even when I'm speaking here, we all bring our biases no matter where we come from. We do not represent countries, we don't represent regions. Acknowledging that, we still have to start somewhere, remembering the shortfalls, but it's time that we start working towards it. Thank you.
Thanks, Senjuti. So we will have another 10-second break to get Ji and Shyam ready online.
Hello, and welcome back to Breakout Session reporting, flagship projects in Asia. We had a very fruitful discussion together with Shyam and excellent panelists, Chung Cha-hong, Pongpan, Aki, Alistair, and Zemin, and Sehan. So I just want to quickly highlight a few of the key takeaways in this slide, and then Shyam will continue with the next two slides. I think it was very important to set the kind of the vision or the direction of where do we see HCA Asia kind of look like in the future. And hopefully, I think some of them have been already addressed in previous presentations, but hopefully the HCA Asia would have achieved a data equity and generate high value data sets, which means that the data that we generate actually have a positive impact to local communities, but also provide the right genomic tools and platforms for local scientists to conduct their own research. We hope that we can also promote through HCA Asia efforts to promote regional science and giving platforms to speak the technologies and excellent science that are being conducted in the region, but also to raise new leaders who can pioneer in the field. And hopefully that will lead to impactful science and publications. And it's also interesting to note that we are the first of its kind in the HCA to initiate a regional effort. And there are other regions in the world who are kind of looking into us for a role model. And in fact, just last month, HCA Latin America had their first regional meeting. And they are really excited to initiate their equity efforts and regional efforts as well. And they're really looking up to our efforts and kind of know-hows to initiate their regional HCA efforts. So I guess we as a community can also suggest and provide ideas and models that can be used for other parts of the world. I think it is important to understand that the genetic diversity, it's a challenging one, but it is highly relevant. Just because even the healthy tissues and the physiology heavily depends on our genetic background, and that's even more pronounced if you study the pathology of disease. So, we can look at the same disease like diabetes, but, just based on your genetic background, how you derive diabetes state could be very different. In a similar token, the treatments and the response to drugs can also vary quite a bit depending on your genetic background, as well as the risks and predispositions of disease based on genetic background. So we hope that it'd be hard to tackle all of this. This is very much in line with genomes and genetics, but hopefully we can address some of these issues through HCA Asia. So you may have heard that, Shyam, Unyang, myself, and others like Chum are part of the Asia Immune Diversity Atlas, or IDA project. And there have been some questions about how to join IDA project. And, we are not any by means exclusive club. We are welcoming any new members, and you're open to join as long as, we are able to generate data locally with harmonized protocols and technology that we've been working on for a couple of years. So that's really kind of key emphasis, is that we would like to involve local researchers and avoid shipping samples out. There was some discussion that in the past in the genome research, the samples were shipped to a data production centers, mainly in the US or in Europe, where the samples were coming from underrepresented populations. And we want to really avoid that and just ensure that the local researchers are able to produce their own data set. In order to join IDA, I guess ethics and data sharing is to comply with HCA policies. And that's really about open data and that it is accessible, especially healthy samples. So we already have many of info package. So they are kind of how to get started on collaborating across different countries. And we can actually use this platform to expand to other tissues and other programs. And this really has been really useful to just jumpstart on how to collect samples and how to standardize technologies and so on. And so hopefully, of course, joining IDA is open, but also requires funding, and we'll be happy to discuss how to work together on building that opportunity. So with that, I will slide and have Shyam talk about the next few points.
Thank you, Jay. Yeah. So then that was IDA 1.0, you can say. And we had some very interesting discussions about IDA 2.0 and IDA 3.0. And thanks to all the panelists and attendees for bringing excellent ideas. So the obvious step is to integrate the eQTL data and chromatin QTL data with genetic susceptibility and derive some biological inferences about which cell types are involved, which gene loci are involved, and so on. And then, of course, expanding. So we want, right now, IDA 1.0 has, depending on maybe 1.5, has 5 countries and 6 ethnicities. But we would like to expand even further, particularly bringing in underrepresented populations and also having larger cohorts to really map the genetics more comprehensively. And for this, we will need training. We will need some infrastructure for training. We'll need more funding, of course. And we discussed a little bit about those issues. And then going from just healthy PBMCs to the next dimensions, which is perturbations. So say some kind of immune perturbation, let's say by drugs or by vaccines, which are very topical these days. And many industry partners would potentially be interested in both of these kinds of perturbations. And then, of course, aging and environment are other perturbations that we're always involved in, interested in. For example, what does a microbiome do to your immunome? What do pollutants do, diet, lifestyle, etc. So if you do have a reference PBMC data set, right, it's very powerful, and there's many directions you can go in based on that. And of course, not just peripheral immune cells, we can compare them to tissue resident immune cells. And we heard about the Atlas of blood cells yesterday. And that kind of initiative is very valuable, connecting tissue and peripheral. And then, of course, cancer blood samples before and after immunotherapy, infectious disease, as Varadam spoke about earlier today. So many new directions for IDA 2.0, IDA 3.0, but then IDA is not the only project we want in HCA Asia. We want other projects as well. Perhaps in terms of genetic diversity, we can look at skin. So that is a relatively easily accessible sample type you can get from healthy cohorts. We can also get iPSCs relatively easily, differentiate them into brain organoids, other kinds of organoids, 2D culture differentiation. So there again, that would be a nice target for genetic diversity. We did also talk about getting autopsy samples, which is a bit harder, but definitely valuable to really map all of the tissues. And then Asian prevalent cancers, for example, gastrointestinal cancers, are particularly prevalent in Asia, and it would be very valuable to have an HCA Asia flagship program on that. And then diabetes is also very, very relevant in Asia. The genetics and environmental influences on diabetes seem to be unique in Asia and unique risk factors. So it'll be very important to characterize that in HCA Asia. So these are some of the new initiatives we hope to start, but cutting across all of these different potential projects, there's some things we have to do, right? One is we have to standardize technology across the different study sites, because all of these HCA Asia flagship programs, by definition, they're not single country or single population programs. They're multi-site programs. So you do have to make sure that there's no technical variation across the sites. You standardize protocols. And then one question that came up there is, if you use the same technology, just one technology, that's like monoculture in food production, right? So it can have some flaws. You might lose some cell types that technology is not very good at profiling. So ideally, if we have the funds, you want to do multiple technologies at each site, but still in a standardized way, and make sure that each of the technologies you use gives you acceptable data quality and also acceptable reproducibility. It cannot be that this data set is great, that is medium, and the third data set is bad, then it's hard to compare across sites. And of course, which technology you're using depends on which biological question you're asking. Is it cost effective? And so on. And you want to push the cost low enough so that it's small compared to sequencing, or at least less than the cost of sequencing, and then cost for the data generation of single cell encapsulation, not such a big deal. And then for any of these flagship programs to work, we need tissue banks, we need tissue resources, and we need modalities for communication. So watch this space. We should have, in a not too distant future, an HCA Asia website that any of you can contact if you're interested, HCA Asia mailing lists, and also mailing lists for specific flagship programs like IDA. And we hope to recruit new programs, new coordinators. For now, if you have more general genetic diversity questions or comments for HCA, you can try this mailing address, genediversity@humancellatlas.org. It's not an HCA Asia specific mailing list, but any ideas for gene diversity for HCA should go there. So I really hope that this fruitful discussion will move into the next phase, which is individual groups of people coming together and offering to lead and coordinate specific projects that can become flagship projects in HCA Asia. And please email Jay and Wong Yang and myself if you would like to contribute to this. Thank you.
Thanks, Shyam and Jay. So now we have another quick break of 10 seconds to get Partha online.
All right. I'm going to report back from the break, which has to do with, or which had to do with ethics and data sharing. This was coordinated by Tim Tickle from the Institute myself. But before I provide to you the report back, I've been asked to talk about a little bit about ethics and equity. Two things that I was supposed to talk about yesterday, but because of certain issues, I missed that. So essentially, I will very quickly summarize the ethical issues. I'm a member of this particular working group that's chaired by Partha. Most of these ethical issues that we discussed in the Human Cell Atlas are pretty standard. One of informed consent and getting approvals from national agencies, etc. But what comes in as something very specific is that in HCAs, because we want these tissues to be fresh, there is some amount of dependence on accident victims, autopsies, cadavers, and so on and so forth. And obviously, the consent on these kinds of cells cannot be provided by the person from whom we are harvesting the cells, and therefore informed consent has to take a different dimension, and that is very specific to HCA, and we talk about that in our working group on ethics. There are also discussions on material transfer and data transfer. There are restrictions on the use of research materials, which is different from one country to another, and we discuss that in the ethical issues. We do have guidance on some of these points, but not on all. There are also various kinds of forms that are being developed such that people who are interested in these kinds of things, either accessing materials or donating materials, will have to fill forms, and that will go through a specific committee. Then there is this issue of intellectual property, because some of these experiments may lead to intellectual property, and they have to be discussed upfront. And how do we discuss those? Those are not always commercial, but there are also ethical underpinnings to intellectual property rights. We talk about data, and we talk about confidentiality of data. I will discuss more about this as I report back from our group today. And then there are countries where they seek a moratorium because they have generated the data, but they don't have the wherewithal to analyze the data and get to inferences in rapid time because of lack of capacity or whatever. So oftentimes a moratorium is sought by the country that's generated the samples, and essentially it's to protect right to first analysis. So these are the kinds of questions that are discussed in the ethics working group. There are various kinds of forms and guidelines that are being developed. There are some that are already in place, such as informed consent, etc. But informed consent from living individuals, which is what is pertinent to Asia right now in the ADA project that Shyam and Jay just described. I will now move over to talk about equity. Equity is another working group that has been kind of spearheaded from the beginning by Musa Malanga, Alex Shelek, and myself. And I will tell you the context of the equity and what are the basic principles that we are discussing in equity. So HCA, Human Atlas, is anchored on egalitarian principles and comprehensive principles. It's first of all compositionally inclusive because we want to make our conclusions generalizable. So it must pertain to gender, age, ethnicity, geography, and socioeconomic status. So we try to cover all of these various diversities such that it becomes compositionally inclusive. And the goal is to provide a cellular atlas or generate a cellular atlas that is representative of humanity such that diversity is also captured. Secondly, it has to be organizationally inclusive. It cannot exclude countries. So look in various countries, and we reach out to various countries. These countries are brought in. There are levels of education. Levels of education are different in different countries, and we have to talk to them, speak to them, and reach out to them. There's training of researchers that we need to do because not all countries are geared towards single cell genomics, which is really the major technology or the basic technology that's being used in HCA. The goal is to be able to identify and overcome political and regional barriers. Everybody needs to be ethnicities, the various levels of SES. So we discuss how best to overcome and identify and overcome these potential regional barriers. There are also, we need to be educationally inclusive, and we need to touch all sectors of society. Again, I will come back to this as I come back and report to you what happened in our breakout session today. We need to educate and be educated by all sectors of society. We have a draft definition of equity. Equity is not something that's easy to define, but a draft definition is first of all, it is inclusive. It has inclusive representation. It has inclusive participation, and it is inclusive with respect to benefit from the data that we generate. So the representation, inclusivity of representation means that we have to sample diversities across age, gender, age, ethnicity, geography, and SES. Just as I mentioned, participation has to be from various such that abilities are fostered. There is leadership in low and middle income countries, and so on. And the benefit that needs to accrue from the data that we generate in SES should not be exclusive, should include in all countries, should span all countries and include all countries. And there has to be a bi-directional benefit, and we have to pay close attention to overcoming existing barriers to data transfer in LMICs and avoid past mistakes. Again, I'm not going to narrate what the past mistakes are in large projects that have been implemented in various multi-country projects. We have actually learned a great deal in the last 2 or 3 years that we have been talking to various people, organizing road shows, organizing phone calls, talking to institutions, talking to groups, etc. And at some point of time, while we were discussing amongst ourselves, we felt that it would be a good idea to put together what we have learned for the benefit of those researchers who are already participating in HCA or those who might be considering to participate in HCA. And this has been done. Our comment has just been published earlier this month in Nature Medicine, and essentially we titled our commentary as Lessons Learned. It's a short commentary. Anybody who's interested can look this up. If you don't have access, just send me an email. I'll be happy to send you a reprint of this. And essentially, the most important point that we have learned is to be patient and patiently listen. I think that's what has been our major take-home message or take-home lesson from the from the discussion that we have held and from the engagements that we have held. Move over and present to you what happened in our breakout session today. I'll skip this slide. This is a guidance that we got from the HCA leadership, HCA Asia leadership, essentially trying to find out what may be the bottlenecks in various countries and various researchers from joining HCA. So I will give you the back. We had a long, lovely discussion. There were not very many panelists or participants, but those who participated were actually very forthcoming and provided insightful remarks. First of all, the different countries have different legal restrictions around metadata and the data which and other kinds of data with respect to data sharing, which is a very complex activity. By the way, this particular summary has been made jointly by Tim and myself. The examples of these complications, some examples were provided to us. For example, the Japanese law requires genomics data to be managed access data. While we talk about open access, there are countries where genomics data has to be managed access. To get access to the data, you have to write to whoever is the owner of the data and get an approval in order to access that data. India has national policies that override any individual or PI consent. So this is important because oftentimes we believe that if the individual who has donated the sample by a specimen, if that individual gives the researcher the right to share the data generated from the sample, then that data can be shared. But in India, we have a national data policy that actually overrides individual consent or even the PI consent. So these are diversities in legal restrictions that we need to keep in mind. This policy is not a legal, Indian policy is not a legal restriction yet, it's a guideline. In South Korea, even collecting comorbidity data, so such as whether a person has diabetes and things like that in the context of SARS-CoV-2, has not been easy because there are privacy issues with respect to collecting comorbidity data. Not only there are constraints on, so these are issues that constrain data sharing. Not only there are constraints on data sharing, but it can also be difficult to convince people to donate data, especially when we are talking about single cell as opposed to bulk data. What we discussed primarily, or what we realized primarily through our discussions today, is that single cell genomic data or single cell transcriptomic data that we are talking about in HCA, essentially even even to molecular biology researchers hasn't sunk in very well. People can think of a tissue and draw RNA or DNA, isolate RNA or DNA from that tissue and analyze that. To be able to do this from a single cell from a tissue is something that still needs to sink in very well. So people are wary about providing data or biospecimens when it comes to single cell analysis. How can we convince others about the importance of sharing biological data? This is a question that came up, and we discussed various kinds of ways and means of convincing others the importance of sharing biological data, such as it will speak to disease, it will speak to state of health, etc. Is there educational material that we can build to improve data donation and also enhance data sharing? So we need to HCA Asia, not just HCA Asia, we've discussed this in HCA organizing committee as well, that we need to provide training material, understanding material, so that it becomes easily understood by people at various levels. Emphasis was laid on imparting education at different levels. In South Korea, for example, people are not so aware about data sharing for research. So why does one need to share data? That concept itself, the panelist who was there from South Korea said that they are not so aware. And the only way to do this is to educate, to develop guidelines and educational materials, consult with people, and popularize these guidelines. This is what we felt. So our committee, or working group, or our breakout group, essentially focused a lot and emphasized a lot on education, development of training materials, and popularizing HCA in various countries such as Asia. And Asia particularly presents a great challenge because of the diversity of languages, because of the diversity of socioeconomic status, and because of the diversity of cultures that there is in Asia. So it does provide a lot of challenge. Then we discussed about the data coordination platform, and there were many technical issues that were raised these have to do with diversities of conception of the way that data are handled in multiple countries. In some countries, for example, there is a dual system, a basic data system which is less private, and a deep data system which is more private. And the data standards also then must have a duality, and these are issues that haven't been thought through very clearly, and these came up in things that was suggested is that can there be some common metadata with some common standards for wider sharing. There was a generous that was shown data, because patient transcriptomics is now easily doable. There was a lot of interest that was shown, but these are high volume data. A lot of it is image data. And so what kind of data sharing are we talking about? Are we talking about raw data sharing, in which case, not easy even to over the internet? Secondly, if these are, then the question came up and the discussion happened that these may be partially analyzed data. So raw data is not what is should be shared or can be shared easily. Partially analyzed data, and to what level of analysis and all of this was discussed. So these were technical issues, and I have not summarized all of the technical issues. Then the final thing that came up was with respect to consent. Who informs of consent to the HCA Asia? Is it the submitting lab? Is it the individual? Is it the nation? And I've already talked to you about differences in opinion or differences in perception of various nations. So the DCP has an upcoming pilot, and this is something that the data coordinating platform is doing, has an upcoming pilot on managed access. If anybody is interested, that's the Wrangler-team at data.humanatlas.org. That's where you can register and you can participate in this pilot project on managed access. So this is what we discussed, and this is the summary of the breakout session that we had. There's a lot of work to be done, but at least we got guidance as to which direction the discussions or future actions must flow, and that was extremely useful, and it's my pleasure to have been able to gotten this opportunity to be able to share these thoughts with you. Thank you very much.
Thank you, Partha, for the brief presentation of the talk that would be expected yesterday, and thanks for the report back of the breakout session. So now we will have a quick break about a few seconds to move on to the plenary discussion session.